Hi, welcome to the Fundamentals at SSRL and LCLS video series. My name is Leland G, and I'm the lead instrument scientist for the MFX instrument at the LCLS. And today in this video, I'll give a bit of an overview of the MFX instrument, its capabilities, and future plans. So the LCLS is situated here at the base of the peninsula of California. The LCLS repurposes part of SLAC's existing 2-mile-long accelerator as part of an X-ray free electron laser with highly spatially and temporally coherent X-rays for use within 2 experimental halls, which we creatively call the far and near halls, based on their relative position to the accelerator. Here's a schematic view of the 2 experimental halls. In the near hall sub-basement, we have TMO, TXI, and XBP. Upstairs are the ChemRix and QRix hutches. Continuing through the X-ray transport tunnel, XRT, takes us to the far experimental halls where XCS, MFX, CXI, and MEC reside. The biology program at the LCLS mostly lives within the MFX and CXI hard X-ray instruments. However, there are applications of utilizing the tender X-ray instrument, TXI, to probe some biological science in the future. There's enough flexibility built into the design of each instrument that parts of the biology program can be accomplished at the other hutches as well, which affords us some operational flexibility. There are other X-ray sources in the world, so why do biology experiments at the LCLS? The key challenges with probing biological systems with X-rays is their inherent sensitivity to damage caused by the X-rays that are doing the probing. In biological crystallography, there's a race between collecting high-quality, high spatial resolution data and that data being corrupted by the X-rays that are doing the probing. At conventional sources, part of this damage is mitigated by using cryogenic conditions and careful radiation dosing strategies, but there will still not be a truly damage-free measurement. With the beam produced by the LCLS, this problem is mostly mitigated by a concept called diffraction before destruction, where the temporal structure of the X-FEL pulses are so short, on the order of tens of femtoseconds, that they can probe the crystal before any of the radiation damage can become part of that measurement. Another added benefit of the damage mitigation is that then samples can be measured under ambient conditions, more closely aligning with their physiological states. The extremely short pulses also enable us to measure ultra-fast dynamical processes in these systems. For example, we can trigger systems with visible light and then probe them with X-rays to monitor dynamics faster than 100 femtoseconds. Likewise, as there is a high spatial coherence from what is basically an X-ray laser, we can focus the beam down to submicron size and probe smaller crystals than what is possible at conventional sources. This really helps with biological systems that just don't want to form large crystals. There are 3 areas of structural biology that the LCLS has strongly enabled. Membrane proteins have historically been difficult to form large crystals of, or crystallize in general, but the development of lipidic cubic phase to form small crystals for probing by an X-FEL has created a research boom for these types of systems. As they are membrane-bound, they typically play key roles in receptor messaging and transmembrane self-transport. Structural dynamics is another strong contributor, which, as I mentioned, we can trigger photochemistry by optical light, but we can also probe catalysis with chemical mixing, and even mix biological systems with relevant gases and probe how their structures enable their functionality. Finally, metalloenzymes, which use the sensitive geometric and electronic structure of metallocofactors to access difficult chemistry, have benefited from X-FELs. Due to the delicate electronic and geometric structure of the metal centers, they are particularly sensitive to radiation damage, with this issue avoided by measurement at the LCLS. And that brings us to the macromolecular femtosecond crystallography instrument, or MFX, at the LCLS. MFX was built during the mid-2010s to meet a growing demand for structural biology at X-FELs and commissioned in 2016. One key lesson from the early days of the LCLS was that biology experiments are varied, and the approaches to tackling biology questions are diverse. We baked this into MFX by making it a flexible, in-air instrument for these experiments. So unlike the CXI instrument, which is completely under vacuum, at MFX we can have a large, in-air sample table where many different end stations can be deployed for measurement at ambient conditions. Another key early lesson was that conventional structural biology, specifically for metalloenzymes, benefits from a multimodal approach where we utilize spectroscopy to simultaneously monitor the electronic structure of the metallic cofactors. This multimodal approach has become the standard mode of operation at MFX. This flexibility and multimodal approach at MFX are key strengths of the instrument. This has enabled us to even expand beyond biology into material sciences, condensed phase chemistry, and allowed us to field high sample throughput programs via mail-in. Collectively, these have led to a number of scientific highlights, including probing transient conditions in samples. Besides the perturbations I mentioned previously, we have also done temperature jump experiments at MFX to see how thermal disorder deposits into dynamical parts of protein structures. Finally, MFX strives to lower the barriers to access in data reduction and analysis for the user community. Here we have developed many pipelines for working up diffraction data and spectroscopy that make these approaches more accessible for users, especially when they're unfamiliar with the high volume of data generated by the XFEL experiment. This is important because SFX experiments at the LCLS are already very complex endeavors. They require challenging detailed work from the sample creation stage, preparing to deliver and trigger the sample, actually collecting the data at the MFX instrument, monitoring that data via online feedback and near-time data analysis, then to finally arrive at a 3D structure for chemical interpretation. So what exactly are we measuring at MFX during an SFX experiment? In a structurally ordered crystal, each layer of the crystal lattice scatters X-rays. Under certain conditions described by Bragg's law, n lambda equals 2d sine theta, we get strong constructive interference of these scatterings. This results in sharp diffraction peaks observed in the forward direction. Using Fourier analysis, among many other tools, we can use this pattern of scattered spots, or diffraction pattern, to reconstruct a 3D structure that generated the pattern. You can find a detailed description of SFX and the diffraction process in the Overview of Serial Crystallography at SSRL and LCLS video in this same series. As I mentioned previously, at MFX we simultaneously measure the X-ray emission spectrum, or XES, while we measure diffraction. In this process, the hard X-ray photons that give us our diffraction patterns also ionize transition metals in an element-specific way. Following this ionization, an outer electron fills the hole in the core and releases a photon. The energy of this released photon is characteristic of the transition metal and the orbital the electron originated from. If it comes from the 2p orbital, it will have the most intensity but least chemical sensitivity. It will encode some information about the oxidation and spin of metal centers by the centroids of the peaks and their full width half-max. This process is called K-alpha X-ray emission. At higher energy but reduced intensity, about 1/8, the 3p to 1s emission line encodes significant information about the metal spin. Finally, at even higher energy, where the electron originates from the outermost valence orbitals, we have the valence core decay, which encodes substantial information about oxidation, spin, and ligand identity. However, the intensity is nearly an order of magnitude weaker than K-beta XES. To measure X-ray emission, we also take advantage of Bragg's law to selectively choose the XES lines based on their energies using specific Bragg reflections of cylindrically bent crystals. These bent crystals take the X-ray emission from the sample and focus it onto a two-dimensional detector in a configuration called von Hamos geometry. At MFX, we use a 6-crystal von Hamos geometry spectrometer that gives us a large solid angle to measure XES from the sample or measure multiple X-ray emission lines simultaneously. Besides X-ray emission experiments at MFX, we can also utilize a dual channel cut monochromator to scan a narrow energy bandwidth beam through the sample. Transition metals characteristically absorb at specific energies, and we can utilize this to measure the X-ray absorption spectrum of samples, which can encode information about the electronic and geometric structures around the metal center simultaneously. For more about X-ray absorption and X-ray spectroscopy in general, please take a look at the Introduction to X-ray Spectroscopy at SSRL overview from Thomas Kroll in this same video series. Due to the polarization of the X-ray beam, spectroscopy at the LCLS must be measured in the vertical direction. At MFX, we achieve this through our exchangeable liquid jet chamber, or XLJ. This chamber has a horizontal mode where the sample is injected horizontally, diffraction is measured in the forward direction, and spectroscopy is measured vertically. The chamber itself is kept under inert conditions of helium to minimize air scattering, maximize signal, and keep oxygen-sensitive samples stable. To monitor sample stability, we employ a number of secondary diagnostics. An oxygen meter attached to the chamber gives us a handle on the anaerobicness of conditions of the sample environment. Crystal vision is a microscope that monitors crystal numerical density and morphology as they travel through sample delivery lines. And an inline UV-Vis system tracks spectroscopic changes in the absorption spectrum to ensure sample integrity. I mentioned previously that there are a lot of different problems to solve in biology, and the approaches to them are just as varied. As such, the different sample delivery options we offer, that is, getting user samples into the X-ray beam, runs a wide gamut of physical states, consumption rates, and specialized applications. At the simplest level, the Rayleigh jet is generated from pressurized flow through a capillary of 20 to 200 microns. This is considered the fire hose of sample delivery. The method is robust and has fewer failure modes than other methods, but the sample consumption rate is untenable for most biological SFX experiments. However, the injection approach enjoys success when the sample can be recirculated, which is possible with most condensed phase chemistry experiments. A step above the Rayleigh jet is the gas dynamic virtual nozzle, or GDVN. Here, instead of driving the formation of the liquid jet with only liquid, we utilize an outer sheath of inert gas with an inner liquid stream. This configuration leads to drastically reduced sample consumption and a smaller jet size, which can be on the order of a few microns or less. This makes GDVNs usually the first option for biological SFX experiments. If sample consumption is an even greater concern, we can utilize the droplet-on-demand system. Here, the sample is dispensed via a piezoelectric-driven actuator in the form of spherical droplets that can have a diameter on the order of tens of microns. These droplets are perfectly synchronized with the 120 Hz XFEL beam, and no sample is wasted during measurement. This system also couples well to high-throughput modalities where the nozzles can rapidly switch between samples during an experiment. Next, we have the droplet-on-tape system, where instead of the droplets being fired and interacting with X-rays in midair, they are dispensed directly onto a rotating capped-on belt. This belt conveys the droplets into the X-ray beam synchronized at the XFEL rate with superior stability and the potential to excite the droplets via gas or laser, then measure at long millisecond delays. Besides liquid delivery methods, crystals can be embedded or sandwiched in various media, and a series of fast-moving motors can raster across the samples with the X-ray beam. The challenge with this method is that where liquid delivery approaches are self-selecting of crystal sizes by what fits through the injection orifices, this fixed target method can introduce extremely large crystals into the beam, which could potentially damage the detector. Finally, crystals can also be embedded within a high viscosity matrix, and with a hydraulic piston, this gel can be extruded out to interact with the X-ray beam. This method, called high viscosity extrusion, or HVE, is often a good option for membrane-bound proteins or for very low sample consumption, but the gel itself contributes a large background, more so than other methods. It also usually delivers crystals at a rate below the maximum repetition rate of the X-ray beam. These methods also have chemical mixing counterparts as well to trigger chemical reactions and monitor their effects with X-rays. Here's a table summarizing their different expected operational parameters. We can see that the colliding droplets approach does have very favorable sample parameters. However, in practice, there are stability challenges with colliding two droplets in midair, and we are actively developing the reliability of this method. More than chemical mixing, the heart of the ultrafast laser at MFX is a coherent Estrella titanium sapphire amplifier, coupled to a Topaz Prime Plus OPA. The system outputs substantial power from visible to the near IR. For most experiments involving the Estrella, a full instrument resolution of about 100 femtoseconds is readily achievable. Further, we have mobile nanosecond lasers for longer delay experiments or temperature jump experiments as well. Beyond cool equipment for the instrument, we maintain a strong documentation base through Confluence. Many operational pages are accessible for users, as MFX has many instances of experienced users independently self-operating the beamline without staff. Further, we have a fully documented Matterport where you can virtually tour the beamline with the various parts annotated with descriptions and links to the relevant Confluence pages. At MFX, we strive to lower the barriers to entry for users unfamiliar with the volume and complexity of XFEL experiments. We've developed automated pipelines for routine calibration analyses through our web interface for experimental note-taking called the eLog. Likewise, when a complex analysis pipeline is prepared, then through the eLog you can view the cumulative electron density you have collected directly within the web interface. These near-term feedbacks are important for users to make critical decisions within finite beam time. As such, we have worked to integrate more robust feedback tools, such as CCTBX from the LBNL team. Here we can monitor key measurement statistics, indexing rates, estimated resolution, and beam parameters in a near-term manner. Our data is also configured to work locally at our S3DF high-performance computing cluster, or at NERSC. These important cross-facility linkages are part of a greater project called Illumine, which develops intelligent modular data workflows with inter-facility pipelines. Likewise, we are working to lower the barriers to X-ray emission analysis through a code package called XSPECT. Since at MFX, X-ray emission spectroscopy is nearly always possible for metal-containing biomolecules, the XSPECT package handles most of the rudimentary data reduction and analysis for these measurements to enable users without XCS experience to use the spectroscopy at least as a diagnostic tool, or with a little more effort as an avenue for additional chemical insights. So we've described a lot of the capabilities of MFX. However, a key highlight of the applications of the instrument's utility is the study of photosystem 2. In PS2, oxygen is generated at the site of a 4-manganese-containing cluster called the oxygen evolving complex. As this system plays a key role in photosynthesis, the reactivity is light-triggered through 4 sequentially light-activated states in what is called the Koch cycle. To understand the dynamics of this cluster under different illumination conditions is key to developing chemical insights into how this photoreactivity is enabled and could be emulated. Here, users from the LBNL lab use their droplet-on-tape system, nanosecond lasers, and our ultrafast laser to pump the drops of PS2 into different states along this cycle, and then using diffraction and manganese K-beta X-ray emission spectroscopy, monitor the geometric and electronic structure of the oxygen evolving complex between all steps of the Koch cycle. With this approach, they were able to map the very fine orchestration of water, protons, and oxygen atoms to and from the OEC up to the point of onset of the final oxygen evolution step. Another example is in the study of isopenicillin N synthase, or IPNS, which is a non-heme iron enzyme that utilizes oxygen for the production of isopenicillin N, which is a precursor for all penicillin molecules. Here, by carefully correlating the iron K-alpha X-ray emission full-width half-maxes with their structural data under oxygen incubation, they could identify the structures exactly consistent with the formation of a ferric superoxide complex that causes perturbations throughout the entire protein dynamical structure. These non-local dynamics highlight the importance of macromolecular structure on the local catalysis within the active site that can be extrapolated to other systems. It also emphasizes that in order to mimic some enzymatically catalyzed reactions, it is likely necessary to have a large macromolecular scaffold to reproduce the impact of these long-range structural interactions on the local chemistry. MFX will also be undergoing an upgrade very soon in mid-2026 along with the LCLS 2 Accelerator. The instrument will return in 2027 as MFX HE. The primary upgrades will be a set of Kirkpatrick Bayes lenses to focus the beam to submicron levels achromatically, and a high repetition rate, large format area detector that can measure single frames at up to 35 kHz. These upgrades will unlock new operational modes for MFX AG, as there it will be able to use more than 2 orders of magnitude of X-rays per second for biological SFX compared to MFX's current performance with LCLS1. This would enable exploration of minor structural perturbations, high throughput enzyme drug interaction screening, and observing rare events occurring in biological systems. High statistical power will enable detailed dynamical studies and low signal imaging and challenging spectroscopic approaches. I hope you found this video discussing the capabilities of the MFX instrument useful. I want to highlight once again that MFX plays a key role in human health research, as well as transformational energy research through extremely complex experiments. We've worked to meet this breadth of research demand by making the instrument modular, flexible, and robust. Despite MFX being considerably smaller hutch than some of the other LCLS instruments, its contributions to scientific fields have been substantial. With that, I thank you for taking the time to watch this video. If you are interested in conducting experiments at MFX or MFX AG, please get in contact with us.